Video, we are going to look at something called a single factor cross. And a single factor cross is when we are mating two organisms, and again, we're going to use plants as our model, and we're really only tracking or paying attention to one character. We're not trying to pay attention to multiple things, just one character. So in this single factor cross, very similar to one we saw a couple of videos ago, we're saying that the parental generation is phenotypically purple crossed with phenotypically white, and they're both pure breeding, meaning homozygous dominant for the purple and homozygous recessive for the white plant. What that means then, from a meiosis standpoint, is that the purple plant can only produce big P purple alleles for its gametes, and the white plant its gametes can only carry the little p allele, because that's all it has to donate. One or the other goes in there, and in this case and in this case, they're both homozygous, and so it's 100% of the gametes for this purple flower are carrying the big P purple allele. 100% of the gametes for this white flower carrying the little p white allele. And so when we cross these two, 100% of our F1 generation are going to be heterozygous, carrying big P and little p both, but they're going to be phenotypically dominant because this is, as we said earlier, a simple dominant recessive relationship. So this is a single factor cross where we're paying attention to only one character at a time. Now you'll hear the term monohybrid cross often. Technically, a monohybrid cross is when we cross two heterozygotes. So it would be like the F1 generation from that cross on the previous slide. Realistically, we kind of use these two terms interchangeably. Monohybrid cross and single factor cross both in practical terms mean that we're just tracking a single trait at a time. But in the strictest sense, the monohybrid cross is when we when we cross two plants that are both heterozygous for the single trait or the single character that we're interested in. So for example, if we had two purple flowers that we knew were heterozygotes, and we knew it because of how we got them. We started with pure breeding parents, and in our P generation, so our F generation is carrying heterozygotes. So the first question we have to ask is, what will the genotypes be of the gametes for both of these organisms? So take a minute, pause this video if you need to, and actually figure out the genotypic ratios for the gametes for each of these two plants. Okay, presumably you hit pause and you scribbled down your best answers. I like to, just so I can remember that what I'm thinking of our gamete genotypes is I actually make little sperms and little eggs out of them, and it helps me to remember that I'm paying attention to the genotype of each of the gametes. Both of them are going to be 50-50 dominant and recessive alleles, so half of each, let's say this is a male plant, or we're looking at the male gametes from this plant, half of the gametes will be carrying the big P purple allele for flower color, and half of them will be carrying the little p white allele for flower color. Always keeping in mind the full complement, the entire genome is present. We're just not paying attention to the entire genome, only to this one particular gene. And then let's say we're looking at the female parts of this plant, and we're looking at the eggs that are being produced. Half of them will carry the big P purple allele, half will carry the little p white allele, and then all we need to do is figure out the different probabilities for mating these two. What if it's this sperm that fertilizes this egg, or what if this sperm fertilizes this egg, or this sperm with this egg, or this sperm with this egg? Those are the only options in that ratio. Simplest way to do that is to create what's called a Punnett square. It's just a little 2x2 matrix where across one side you put one set of the gametes, and across the other side you put the other set of gametes. Then in each box you put you want to enter the genotype of the zygote that's produced upon fertilization. So if half of the male gametes are carrying the big P allele, then half the time you expect that it's going to be the one that fertilizes either this egg or this egg. Right? Half and half. And so if it fertilizes this egg, what genotype do you get? If it fertilizes this egg, what genotype do you get? And so what we see is that if a big P male gamete fertilizes a big P female gamete, we end up with a homozygous dominant zygote. If, however, it's this sperm, the other half of the options for this male plant, then it's carrying the little p allele and it fertilizes the big P egg, then we get a heterozygote. And again, big P fertilizing with little p, we get another heterozygote, or little p fertilizing little p, and we get a homozygous recessive. So what we see in our genotypic ratio is 1 to 2 to 1, homozygous dominant to heterozygous to homozygous recessive. 1 to 2 to 1. Phenotypically, what we would see is 3 to 1, dominant phenotype to recessive phenotype, because big P gives us the dominant purple phenotype, the heterozygote does as well. So what we would see would be 3 purple plants for every 1 white plant, roughly in that ratio. Now, if you only got four offspring, you may not see that precise ratio, but if you've got hundreds or thousands of them, it's going to become awfully close. Now, one of the questions that this brings up is, what if you're just looking at your plants and you see you've got 3 purple offspring? How do you know if they're homozygous dominant or heterozygous? What can you do? Well, we can do something called a test cross. We want to know, based on a dominant phenotype, whether we're carrying a heterozygous, genotypically heterozygous individual, or if it's a genotypically homozygous dominant individual. What would you do? How could you experimentally figure that out? Maybe hit pause and think this one through for a minute before I show you the answer. The most straightforward way is to take your dominant phenotype plant and cross it with a recessive phenotype plant. Well, why is that? Well, we know the genotype of the recessive phenotype plant because it has to be the homozygous recessive genotype. So, if your purple flower is coming from a homozygous dominant plant, then when we cross it with a homozygous recessive plant, in other words, it's got the recessive phenotype, we're going to get one set of ratios of offspring. If, however, our purple plant is actually heterozygous and we made it with homozygous recessive, in other words, the recessive phenotype plant, we're going to get another set of ratios. So over here on the left, if our dominant plant is homozygous dominant, then we're going to get 100% heterozygotes, aren't we? Which means no matter how many times we mate these two, we're always going to get the dominant phenotype, right? However, if we've got a heterozygote as our phenotypically dominant plant and we made it with a phenotypically recessive plant, okay, I got ahead of myself. 100% will be heterozygous, 100% dominant. But over here, we're going to end up with a different ratio, aren't we? We've got 50% or 1-to-1 heterozygous to homozygous recessive, which means we're going to get 1 purple plant for every 1 white plant. So about 50% of our plants are going to have the recessive trait visibly as a phenotype. So all we need to do is take our dominant, phenotypically dominant organism and make it with a phenotypically recessive organism, and we have 2 different expected outcomes based on whether it is homozygous dominant or heterozygous. That's what we call a test cross. Make sure the test cross makes sense to you. Think it through. Let me know if you have questions. I think that's where I'm going to stop this video, and we'll pick up where we left off on the next video.